You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I have Tony Altar, Senior Scientific Fellow and Advisor at Verge Genomics. Website is vergegenomics.com. And we're going to be talking about brain illnesses and how genomics and gene therapies can provide new mechanisms of treatment. So thanks for coming, Tony. I appreciate it.
Richard, it's my pleasure to be here, and I really would like to thank you and Future Tech Health.
Yeah, no problem. So tell me, what got you interested in this area of science? Why not something else? What fascinates you about this?
Well, I became interested in the human brain and what can go wrong with the brain to cause disease when I was a 17-year-old high school student in West Los Angeles. You know, around that time, there was a lot of use of psychedelic drugs, and it was actually really fascinating to me to see the use and effects of those compounds. You know, it seemed that if a single molecule could create temporary psychosis or euphoria or mood disorders, you know, could then the mechanism of psychiatric illnesses share these same features? Could they have a similar single or even possibly simple cause? So that was really fascinating to me and my contemporaries around that time. You know, luckily I'd finished all of my training at high school before the end of the normal period, so I was able to go and work as a volunteer at the Brentwood Veterans Administration laboratory. It was actually the Neurobiochemistry Lab, which I'm not sure I could even pronounce back then, but it was fascinating to me. I only understood, you know, some of what the guys in the lab said, but they took me under their wing, and you know, I learned a lot, including the scientific methods to study the brain. You know, I was lucky to have a desk that I could sit at, and one of the little poster that was on the wall by the desk described how understanding the cause of disease is the first step to finding a cure. I think it was from a drug company promotional brochure, but that became like a mantra for me, and it really has for, I think, all medical innovators. This message really hasn't changed. But the question then is, so how best can we understand the basis of disease, especially psychiatric or neurodegenerative diseases, which are, you know, in the brain, it's not very accessible, it's hard to study. So really the question was, what are the best methods, what are the best approaches that we can use that will pay off and help guide us to helping these patients? So that was
there's a lot of different, quick question. There's a lot of different, you know, neurodegenerative diseases. Each of them is so vast in scope. So have you focused in on one or two conditions, or what's your focus?
Yeah, I have in my career, and at Verge Genomics, we're really focused on amyotrophic lateral sclerosis, or ALS, and Parkinson's disease. Those are the first two. Starting to study schizophrenia a bit as well. And, you know, the mission at Verge is to understand what causes disease so that we can convert that knowledge to new therapeutics. But, you know, really, Richard, in practice, not just, well, before today, it's been a 10 to 20 year step each way, you know, understanding the disease mechanism, and then maybe another 10 or 20 years to come up with the right drug. And this is a long process in practice. But the two things I'd like to talk about today, genomics and gene therapy, I think are going to broaden the scope of diseases that we can address. and also hasten the period of time it takes to develop therapeutic entities.
So genomics, is it studying genes and gene expression? Is it more than that with epigenetics and how do you define genomics?
Yeah, genomics in my world is a little more narrowly defined as to the changes in the expression of genes in the diseased tissues. The epigenetics is another level, how the genes are modified by methylation and acetylation. And that is another interesting approach. It's a little even earlier, I think, in terms of development, but something of interest in the future. And we're really focused on, mRNA changes. And the reason for that is because, if you have a set of, in this case, brain tissues from patients, let's say in Parkinson's disease, we study the substantia nigra, which is the part of the brain that dies in Parkinson's. You can look at the gene expression in those patients versus people who died at similar ages but not with Parkinson's disease. You can start to get what is called a disease signature. That is the collection of genes whose changes are predictive of the patients and don't change in the control patients. And that's really my definition, a broad definition of genomics, is that mRNA change.
Well, I don't know if this is like a curveball, but you said earlier you saw a saying from a drug company saying they need to understand the cause of disease. But what if the cause of disease falls outside of the narrow window that you're looking at it in, let's say? I'm not saying narrow bad. I'm just saying that's the choice of Verge Genomics. Why? What happens if it has nothing to do with mRNA? What happens if it has to do with some other mechanism? Like it may be overlooked, for instance. So do you feel like the approach is valid, or it should be wider? Or?
Well, the net that we cast actually in terms of profiling the mRNA is about as wide as it can get. There are 20,000 genes in the human body. The brain is actually a little bit unique. It expresses almost half of those, so it's got a very rich transcriptome. The other half of the genes aren't neuron necessary, so they don't bother to make the proteins that are encoded by those genes. And, we know about the 20,000 genes because that came from the work of, the Human Genome Project, which concluded from NIH and Celera Genomics back in 2000. So now we have those 20,000 genes, so we can study essentially all of them. And for maybe the listeners who aren't familiar with the DNA mechanism, those 20,000 genes, each one of those codes one or maybe several different proteins, and that's what makes a liver cell or a muscle cell, kidney or skin cell, that type of cell, because they make a particular subset of those 20,000 gene products. And as I mentioned, the brain has a pretty rich one. 8,000 or 9,000 of those 20,000 genes are found in brain cells. And so you can measure the activity of each of those genes by simply measuring the amount of mRNA that's produced in the diseased tissue, and then comparing that with another group of tissues from patients who died but without the disease. And so we do cast a broad net. We do end up with what we call an mRNA disease signature. Your point's a good one, Richard. Certainly we're hinging our bets on the fact that the mRNA changes are revealing the underlying pathology of the disease. And there are plenty of examples where that is not exactly true. So, for example, the mRNA could be made at the same normal level, but it could have a mutation in it that causes that mature mRNA to actually make a dysfunctional protein. And that may not be picked up by some of the mRNA profiling techniques. So it is one method. It's not the only method, but it's a pretty powerful one, as I could describe for a couple of these indications.
So starting with Parkinson's, you mentioned that it happens, I guess, in the substantia nigra. What have you observed appears to be the cause of it, and what's happening, and how does the progression occur, and how does it hurt the person?
It's interesting that you asked about that, because one of the things that we've been doing at Verge is using a very new technique called single nucleus sequencing, where we have actually looked at first several patients with Parkinson's and several controls using this technique in the substantia nigra. What single nucleus sequencing does is it allows you to measure the mRNA in the nucleus of each of the cells in a diseased brain area. So instead of getting an average, let's say, of the mRNA for each gene, but in a dissected tissue, which is unfortunately a mixing of all the cells and therefore kind of an averaging effect, instead with single nucleus sequencing, you can get the mRNA measure for each gene in each cell. So it's a spectacular advance that's just come to the fore in the last few years. So, for example, we've dissected out the substantia nigra and profiled that using the single nucleus sequencing method. For the thousands of cells that we had in each dissected area, we were able to then relatively easily categorize each cell as whether it's a dopaminergic cell originally, or whether it was a GABAergic, or a microglia, or an astrocyte, and about 10 other different cell types that we found in our clusters. And so what you can do now is you can actually take a dissected brain area and look with single nucleus sequencing at just, let's say, the dopaminergic subset of cells, and then we can study the genes that are different in those dopaminergic neurons in the patient versus the controls. So it's kind of an amazing way of separating out the cells and looking at all of their the gene expression as that uniform population of cells. So we're really comparing apples to apples as we make this kind of analysis. And one of the things we've discovered already, and this is a very preliminary pairings of patients, we're going to do quite a few more, but we've already discovered a clear deficit in iron metabolism, which reinforces some of the more compelling among many theories about the cause of Parkinson's disease. So it helps you focus your research, especially if it overlaps with what's already been published, but it allows you to select from multiple theories and really focus on one or two that your own independent analysis helps to reinforce.
Would you, I wonder if it would be possible to take imaging of a small area of the brain, that you want to study, then you're doing this single nucleus sequencing and literally map out visually where the changes are occurring and how they're occurring. Are they spreading from a central point? Are they uniform? Do they have a certain character?
So absolutely that can be done. The caveat is it's not usually, and probably has never, well, it's rarely done in the same patient. There are examples in Alzheimer's where people have been able to image the amyloid plaque burden and how it grows over time in one patient, and then that same patient has died, and one can then go in that same brain and do an analysis. I don't think that's ever been done for single nucleus sequencing, although that's an interesting potential. But imaging does let you zero in on the areas that change earliest in disease. Those are the ones we're typically interested in. And then in other patients, we can then take the brains and dissect out just those key areas and then continue to study them with these different techniques.
So like in Parkinson's, is there an area of the substantia nigrans that first is affected? Does it happen to the center of it, the side of it? Are there other structures that are adjacent to it that might play a role?
That's interesting, and our own data already helps us answer that question. We already knew that the pars compacta of the substantia nigra, which is where the dopaminergic neurons live and then eventually die in Parkinson's, that's the key area. And interestingly, we'll be able to see in our broader study whether any of the other cell types are affected. There are GABAergic neurons there and other cell types. And so you can really look at how specific is this disease to that one neuron type, or are there changes that are going on throughout the brain area, and that those particular vulnerable ones just happen to be bystanders that get killed, or is there something special about them that makes them vulnerable and that they die? And we already have evidence that latter is true, that the dopaminergic neurons have certain components that really make them more vulnerable. And we'll know more as we do more in this particular area of research, but we're learning a lot with this particular technique.
Have you identified how Parkinson's appears to start? Even in looking at a healthy brain, a control as you call it, have you seen any evidence of Parkinson's starting? Does it exist in all brains or only certain ones?
Well, in fact, it does. That's an excellent question. Unfortunately, dopaminergic neurons, for certain reasons we're just beginning to understand, are generally vulnerable. Roughly 0.5% of your dopamine neurons die every year of your life. So if someone lives to be 90 or 100 years old, they've lost up to half of their dopaminergic neurons, just naturally, without necessarily having a genetic or other specific cause of Parkinson's disease. Fortunately, there's redundancy in the system. So if you've lost just half, you might have maybe a few subtle effects like longer reaction times, things like that, but not any frank tremors, rigidity, or other symptoms of the disease. Interestingly, there's another population, serotonergic neurons or serotonin neurons, that die even earlier. And there was a recent imaging study, actually just a couple weeks ago, that showed that you could detect the loss of those serotonin neurons in the Parkinsonian brain by PET imaging well before the Parkinson's symptoms appeared. And this is really consistent with the fact that serotonin and other populations of neurons are actually quite vulnerable as well. A lot of emphasis is placed on the dopamine neurons, but there are other cell types that are just as vulnerable and maybe even earlier vulnerable neurons that die in this disease.
Very interesting.
The reason there's a Parkinson's disease is you can treat the patients with L-DOPA. That replaces the missing dopamine. It doesn't replace the missing serotonin, and the patients really get noticeably much, much better. That's part of why there's been such a big emphasis. And dopaminergic neuron loss was easily detected. The cells are actually dark. When you do a dissection of a brain from a patient who recently died, you can actually see the dark cells without any aid, just visually. They're just clearly visible. In Parkinson's, they're clearly and visibly gone. So that directed a lot of attention to these dopaminergic neurons as being the first ones discovered.
Well, now that you know the serotonin-related ones are also affected earlier, it makes you wonder if you coupled L-DOPA with SSRIs, that would have a worse synergistic effect.
And in fact, that's absolutely done because these patients often have depression. And that's not just because they have Parkinson's, but they're probably depressed also because they're losing the serotonin neurons. So they are commonly treated with SSRIs, and that does help with their depression. So the real challenge here is how can we stop these cells from dying? And that's where the genomics work goes in, learning about these pathways, like, let's say, iron metabolism. We continue to find that as a cause. It'll direct our interest to drugs that prevent excessive iron oxidation. And that's the whole idea behind genomics, that it really helps you identify the drug target. And it may be a totally new target, or it may reinforce one that's already known. We've done the same at a company called Psychiatric Genomics that I helped establish and run in the early 2000s. And talk about being on the bleeding edge, we were definitely there. This is just when the human genome was first sequenced. And so we boldly went on to looking for gene mRNA changes that could tell us about the cause of schizophrenia, bipolar, and depression. And we did the same kind of gene expression profiling compared to healthy age-matched controls. And what was really interesting is that we were able to discover that in schizophrenia, but not in depression or bipolar, we could find a set of genes that changed really quite dramatically versus healthy controls. And we replicated those in another cohort of patients. And so that gave us a large set of genes whose decreases in schizophrenia were confirmed. We were able to look at those genes just as a list of the changes, and it really became quite clear without any complex artificial intelligence, just using our natural intelligence. We were able to look at the list, and it was clear that these were genes involved in cell metabolism. And this was consistent with what was already known, again, by imaging, that in the brains of patients with schizophrenia, there's a decrease in glucose utilization and brain metabolism. So what was really amazing to us is other people had done some similar gene expression profiling studies in patients with diabetes, looking at skeletal muscle. And what they found was the very similar set of the genes. In fact, the two lists were almost overlapping. Genes that were decreased in the schizophrenic brain were also decreased in the people who had diabetes in their skeletal muscle. And so this confirmed our hypothesis that perhaps schizophrenia is like a diabetes-like condition of the brain. So we proposed that insulin or some insulin-like protein, of which there are a few known, and are both in the brain, that their function is diminished in schizophrenia. And this helped us identify a new target for schizophrenia. It's actually a receptor, which had already had some clinical validation in patients. But the significance was really not as well appreciated until we made this discovery that this receptor target can replace the missing insulin signaling to correct the gene defects that were seen in the brain of schizophrenic patients. And we actually showed in cell models that we could reverse those very same gene deficiencies with these receptor ligands as well as with insulin. And so it's a very exciting concept. The reason it's exciting is there's really no other target for schizophrenia except for the dopamine receptor blockers that are common to every single antipsychotic medication that's out there. So this heralded a new, potentially non-dopaminergic target to which several drug companies right now are working to come up with novel ligands to stimulate that same receptor and potentially treat schizophrenia and related forms of cognitive decline. So it's an
Why do you,
interesting.
why do you,
Mhm.
yeah, quick question, why would iron metabolism change? Why would glucose metabolism change after, 30 years of the same brain, 50 years of the same brain? And what about the underlying metabolic pathways? Is there something
Yeah.
to be found there, or do you think it's because of changes in genes and gene expression mutations?
Yeah, let's start with the iron metabolism in Parkinson's disease. I mean, the changes can be very subtle because, as I mentioned, there's already a 0.5% loss of your dopamine neurons every year just normally. That might itself be due to iron oxidating reactions. Iron catalyzes the production of free radicals and oxidative species. That's well known. It's probably true even in normal brain. There are protective mechanisms that protect against that, but maybe not quite enough. And so the cells still die, but at a slow rate. All it takes is a subtle weakening of that protective capacity. And now this iron catalyzing reaction is greater in a Parkinsonian brain. Instead of a 0.5%, maybe now patients lose 1 or 1.5% per year. So after 50 or 60 years, the neurons are mostly dead. And your question, that still doesn't answer your question, though, which is a really good one. What other mechanisms are causing those systems to become more active? And so we discovered in this very preliminary study that a couple of the important proteins that allow iron to be ferreted out of the cell are diminished in the Parkinson brain. And so you could imagine that if iron is accumulating more in the Parkinsonian neuron, that iron can catalyze these toxic reactions and kill the cell. So we're perhaps just on the mechanism that creates the disease. And that is very exciting because now you've got a specific hypothesis that you can start testing and entertain others. I mean, there are probably some others that will come out of our more expanded studies. And then it becomes a matter of doing in vitro and in vivo testing with model systems to see which of those really continues to be convincing. And then from that point, you get more focused on your target and your drug discovery effort.
It's weird. It's like an endless chain of whys. Why is this? Well, then why is that? Well, then why did this happen to cause that? It's hard to know when to stop and at what points the intervention would be useful. But I guess you want to intervene at the first step if you can that you discover, so you don't have to endlessly dig to things out.
Well, that's exactly right. And that's been the conundrum in drug discovery, certainly in central nervous system diseases. We really don't have the full confirmation that we had the right target and the right drug for that target until we've gone through very expensive and extensive clinical studies in patients. Then you get a good sense, if you have a good effect, that you were right. You still have the broader population of patients after the drug is approved to know that that was still a safe as well as effective approach, but it's a long process. The reason that that's interesting is there's the other half of what I was hoping and looking forward to talking about today is the second big revolution in treating brain diseases after genomics, and that's gene therapy. So gene therapy gets to your very point, Richard, and that is, why not just go right to the cause of the disease and correct that? And for all these diseases we've been talking about, certainly ALS, Parkinson's disease, and many, including Alzheimer's, there are in subsets of patients a well-known genetic cause for their disease. So there's a genetic mutation for each of those that predicts that the patient will get the disease with 50, 75, or even 100% certainty. Fortunately, fortunately, this is in a relatively small percentage of patients. And the reason I say fortunately is these tend to be more severe forms of the disease, and they tend to occur earlier than the sporadic or ones that we really still don't know what the genetic cause is. But for those, there's still hundreds, thousands, and in some cases, tens of thousands of patients with these gene mutations that gene therapy really can address. So now for the first time, and in fact, only as recently as 2017 was the first FDA-approved gene therapy in existence in the United States. So only since 2017 have we an FDA-approved drug that really can get at the cause. And this is a drug called Luxturna, developed by Spark Therapeutics, and it was the first approved full-length gene therapy, in this case for a form of blindness. It treats people who have a real problem in low light vision, and it was a tremendous excitement to the community to have this approved after intraretinal injections of this were found to treat essentially almost every person that got the therapy. So
well, that's what I was just about to ask you. You don't want a therapy probably systemically acting. You want it locally acting, but getting into the brain is not so easy. So how would you do gene therapy in the brain?
Well, interestingly, neuroscience comes out as a lead in another area of medicine where you might not have expected it to. It's hard to imagine that we're going to cure blindness, but we have with drugs like Eylea from Regeneron and now this gene therapy approach from Luxturna. And there are others. Spinal muscular atrophy is another CNS-associated disease. It was approved for Biogen. It's treating spinal muscular atrophy. Interestingly, another drug that's on its way in the gene therapy areas from Voyager Pharmaceuticals. It's a gene product that increases the levels of an enzyme that actually converts L-dopa to dopamine. And by injecting that directly into the area of the brain that needs that dopamine, Voyager has been able to show that they can greatly decrease the amount of L-dopa that patients need, and that the efficacy of the L-dopa that they get is increased. So it's interesting. The brain is not a very accessible area, and it is true. If you had to inject these gene therapy products intravenously, some of them look like they might get into the brain in a low level, but their effects are greatly diminished and diluted by going into the blood. So the direct brain injection has been the preferred route. It'll be interesting to see other applications. In fact, the same enzyme that Voyager developed, another group has been able to inject that into the brains of kids who don't have that enzyme, and their dramatic mood and other cognitive disorders have been corrected by that kind of therapy as well. So perhaps, I was going to say that perhaps the limited spread of these agents when delivered to the brain has been partially a blessing, that it limits the therapy to really where it's needed and concentrates the gene therapy product to where it's needed.
Very tricky stuff.
Well, it is.
Oh, well, I haven't gone into the mechanics of gene therapy. So are you doing experiments where you're doing like gene knockouts to see the effective genes? You know which ones need to be changed, or you're looking at more of the expression, and that's telling you more directly which genes would need to be affected to make a change?
There are a variety of approaches in the gene therapy area. One of the earlier ones was to inject or treat with what's called a cDNA, which is really a copy form of DNA that now can make the full-length protein. The problem with that is many genes are too big to be delivered by cDNA. They actually just can't fit into the adeno-associated virus, which is commonly used to deliver them. And so that limits that approach. Another one that we've all heard a lot about is CRISPR, which is a little bit complex in the sense that it needs multiple reagents, an enzyme, as well as the CRISPR reagent itself to correct the DNA mutations that exist. That fixes the DNA, and that there are advantages around that. But there are other approaches which I think are even more exciting that can actually edit the mutations in the DNA. That edit mutations that are existing in DNA, but after they're converted. So, before DNA or deoxyribonucleic acid is used to make a specific protein, it has to be copied into what's called pre-mRNA. So that's the same copy. It's actually an exact copy of the DNA code, but the nucleus makes it in the form of ribonucleic acid. And this is called pre-mRNA. So pre-mRNA is, as I said, a complete copy of DNA, but it contains all the introns or intergenic regions as well as the exons that are the coding regions that make the mature protein. So this intergenic region, the intron, has to be removed so that the gene coding exons can be spliced together. This reaction is called splicing. It occurs in every nucleus of the cell, and it forms that mature messenger or mRNA that we discussed earlier when we talked about genomics. So almost all genes need that form of pre-mRNA splicing. I think it's something like 95% of all genes are spliced like that. And it's really when you think about it, it's the dominant natural form of editing to mRNA and causing the correct proteins to be made. So it's really not surprising to think that, if this is the natural dominant gene editor, perhaps can we exploit that process and hack into it, to correct the gene mutation. And in fact, that has occurred. It's a process called transplicing. And I've been reading up on this quite a bit. It's been done in over 30 examples of disease gene correction in both human cells and animal models. It hasn't gone to the clinic yet, but I think RNA transplicing will become if not the dominant mode of gene editing, because it's already been shown in these cell and animal-based models to be applicable to a large range of diseases. So the idea is, if you inherited a gene mutation, let's take an example, Huntington's disease, where we know the cause of Huntington's is a repeat of what's called a CAG sequence. If you could convert that CAG repeat to the normal shorter form of fewer CAG repeats, I think everybody would agree that you would essentially cure the disease, because the only difference in people with Huntington's is this one mutation. So what splicing molecules have been shown in some preclinical studies in animal model in human cell line, that you can actually convert that very long Huntington creating CAG repeat into the much shorter form of the gene. And as a result, the protein is corrected and the cells live much longer, and they have higher metabolic rates back towards the level of normal, and they live much longer. They don't show the programmed cell death that occurs in the Huntington's cell model. So here you have an example where you haven't had to touch the DNA, but in only those cells that express the Huntington protein, you are able to correct that Huntington gene, not in every copy, but in many copies, to form the healthy form of the gene. And as a result, the healthy form of the protein. And as a result, give evidence that this could be an approach to be used in people. So that's a form of gene editing, which I think is quite exciting. And it's got examples of many other conditions, including tauopathies, hemophilia, cystic fibrosis, and the list goes on and on.
This may be ancillary, but since you can do single nucleus sequencing, has anyone looked to see if there's microbes in the brain, like a microbiome of the brain, or viruses that are in there that could be affecting these conditions or modulating them, and then thereby sequencing the microbes found, if any?
That's a possibility. That's kind of outside of my range of expertise. I know there's a lot that's been said about the possible conveyance of viruses or bacteria, actually, from the gut through the vagus nerve into the brain, explain diseases like Parkinson's. I just haven't really studied it that much to give a good answer.
Okay. Yeah, I just thought it came to mind. So what are some of the milestones that you'd be super excited to achieve, the next few years with your work?
I think personally, for us at Verge Genomics to continue in our ALS program, where we've already produced some really very potent molecules that are active at the target we've identified for ALS, to see that go into a clinical development plan and actually help patients. We've talked to people in the ALS community. Patients are really excited to be enrolled in these kinds of studies. So that would be a terrific milestone. Another one in our Parkinson's area would be to identify targets that have been validated in the literature or potentially completely unknown, and be able to create a drug discovery program around them and validate them in cell models as well as in animal models. And there we have there are a lot of good models available for what happens after the Parkinson's is developed, and even some pretty good ones that create the disease and may do so through the natural disease creation process that mimic that process. So, you can get a reasonable level of confidence that your drug is going to work. And so I think with the right evidence, Verge is well positioned to have compounds that show real promise for Parkinson's and could well be in the clinic for ALS in the next few years. So personally, I think at Verge, that would be a real goal. I think in the industry, I think there's a lot of reasons to be excited about where gene therapy and genomics are going in the next few years. And I think there's going to be real growth in companies and maybe even in the government services for sequencing people's DNA, even at birth. Until now, there really hasn't been as much need because there hasn't been so many actionable results of knowing that you have a Huntington's gene mutation or knowing that you've got a presenilin mutation for Alzheimer's disease. But over time, with the advent of new therapies, there will be things that we can do to actually address this. So I see a growth there. I see a tremendous need for companies that analyze mRNA in patient samples at the single cell level. I see a growth in companies that develop drug and gene therapies based on these discoveries and that manufacture the vectors that are needed. Already there's a 2-year waiting list for companies that manufacture adeno-associated viruses, and that's common at many facilities. There's also going to be a growth for genetic counselors, because the more genes that we can address through these approaches, the more it really is useful to know if a person has, what are the gene mutations that a person has, because now there's maybe something that can be done for those. So that's another area of real growth I see.
Okay. Well, very good. And what's the best way for people to to read papers that you put out, or to get in contact with Verge, or ask questions?
Well, the Verge website has got some information. There's a society that I recently joined, the American Society for Gene and Cell Therapy, which is for those who are more technically associated, the scientists on the call. I think you'd really be excited to go to that meeting. I came away from many lectures impressed by the progress, and then by the end of the meeting, I'd come away a little disappointed if I didn't see a group of patients who were now living that otherwise wouldn't, or animal model that worked phenomenally well where previously nothing had worked. So that's a great society for the progress in gene therapy. That's another way, and there's just a lot happening in the lay press as well. I think there's a second wave of all this to consider as we learn about the genes that are really impactful and the targets that are validated by gene therapies. That also now opens up another wave of pharmaceutical discovery, because we've always been looking for targets in the pharmaceutical industry. And if we can confirm through gene therapy that a certain target is really the target to go after, the drug approach now becomes a lot more straightforward. And with more confidence, we can address drugs for that particular target. To date, that's been a big, risky thing. But I think gene therapy will actually enable the pharmaceutical industry. And conversely, that's true too. If we know that certain drugs work for a disease, but we're not getting complete efficiency, if we know that pathway is the one that's important, but the gene therapy doesn't quite do the trick, there may be a drug therapy that can be used as well. So this will open up the area of repurposing medications that are already known for other indications. We can now take those drugs, knowing what pathways they affect, and try them for diseases for which now the pathways have been better elucidated through the genomics work and potentially even the gene therapies that work. So, there's going to be a lot of crosstalk between these fields that I think is going to make a big increase in the breadth and the impact of drugs for the future.
Yeah, I definitely hope so, because these are serious conditions that affect a lot of people in a terrible way. So, well, very good. And Tony, thank you for coming. I appreciate it, and your time on the podcast.
Richard, thank you too. It's a real pleasure to be able to share what I think is a fascinating area for your listeners. I think just to leave your listeners with a vision that I think the future is bright for treatment and even cures for diseases that, maybe 10 years ago, we'd be hard pressed to see or even use the word cure. Clinical proofs take time, and that's the hardest, longest part of this whole process. But I think this greater breadth of diseases and the shorter time to develop particular therapies for them is going to be a result of what a growing revolution in genomics and gene editing.
Thanks.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier, Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.